We are continuing today with hormones, and we'll lay some foundation to understand cell communication before we dive into the main thing about hormones. Now, in the human system, we use the human system as the example that because most of the things we are doing is about the human body. So we're talking about cellular communication in the human system. Of course, some of these you find also in some animals. Now, there are two major forms of communication in the body, and that is by chemical communication. Some biomolecules are secreted by some glands, and these chemical compounds, these biochemical compounds, stimulate some biochemical responses. And then we also have electrical signals. These move along nerve cells. So these will be in the nervous system. Of course, the biochemical molecules that are secreted as the first group that I mentioned, even that, when they simulate some biochemical response, and they say there should be some nerve signals that are moved between, the synapses. At the synapses, you still have, neurotransmitters, which are still biomolecules, which will transmit these electrical signals from one nerve to the other. So you have these two forms, these biomolecules, neurotransmitters, mostly proteins, which are responsible for communication along the nerves. And then you have what you refer to as the endocrine system, where you have some tissues which will secrete some biomolecules that will trigger some biochemical response. They will stimulate some other glands, or they are going to stimulate some tissues, and the cells of those tissues will respond by some reactions. So we'll take a look at different forms of the responses to have a good understanding of how hormones work. Simply, if you think of it in general terms, that even among us humans, say organisms, if there is no way to communicate, you can imagine how hard that is going to be. Nothing at all. Like there's nothing like language that you can use to tell another person anything. Not even sign language. It will be total chaos. Everyone will be just an individual somewhere, whatever it is that is happening to you or to something else you would like to communicate. There's just no way to communicate. So communication is very vital, even at a cellular level. That yes, if you take multicellular organisms, cells communicate among themselves as a way of survival. So for instance, if you have some intrusion or some invasion of some bacteria, some virus, there should be an immune cell that recognizes such an invasion and then responds to it. And its response actually communicates to the other cells. So everyone, all the other, immune cells and any other cell is also prepared to fight such an invasion. So it ensures survival. The cell communication ensures survival, apart from it producing biological responses such as the processes of transcription, translation, which will be the synthesis of some specific proteins, which will be enzymes, neurotransmitters, which will eventually cause some biochemical processes to respond to any either internal or external stress. Now, even with single cellular organisms, that so for instance, in the SS, we did things about some protozoa, some flagellates, paramecium, and things like that. You realize those organisms, as single cell organisms, also have their form of communication with the environment. So they are able to recognize some danger, some stress in the environment. And if they have to change their course in a different direction, they do so to prevent any harm. Now, if you take any two cells, that one is a signaling cell, that is a cell which secretes the biomolecules. So what you see on the left-hand side is a signaling cell which is secreting some biomolecules, which we are referring to as ligand. Now, so a hormone can be a ligand. The hormone is a ligand. And you have a target cell, a tissue that has some specific cells in it that will recognize these ligands. A target cell will have in its cell membrane what we refer to as a receptor. The receptor can be a glycoprotein, a glycolipid. So it's a complex of the biomolecules that you have studied already. Carbohydrates, proteins, lipids. So usually you have these receptors being some transmembrane proteins, can be some transmembrane glycoproteins, glycolipids, or lipoproteins that have interaction with the ligand. And when they have interaction with the ligand, they send a signal into the cell. And we'll see some mechanisms as we go on by which some transcription is sort of stimulated in the nucleus for some specific proteins to be synthesized. Or maybe the ligand itself, if it is lipophilic, because you know the cell membrane is phospholipid bilayer. And so if the ligand is soluble in lipids, then that ligand will be able to pass through the cell membrane and go into the cytoplasm, bind with a receptor inside the cell, and then will activate some biological response to whichever stress that it is responding to. So you have two cells, communicating in this manner. That's a general way to present how cells communicate. Now, there are different forms in which cells communicate. One of the forms we refer to is the autocrine signaling. Now, the autocrine signaling you find in the immune system mostly, that a cell secretes a signaling molecule, and that signaling molecule is actually signaling that cell itself. So for instance, the diagram you are seeing on your screen, you see that the cell, number one, you have number one in the cell, the cytoplasm, where some signaling molecules are secreted. So number two, you have the signaling molecules out of the cell, and these signaling molecules bind onto receptors on that cell itself. And the response will be that there will be an activation of a gene in the nucleus, so that gene is transcribed and translated. So you have a protein that either it's an enzyme, neurotransmitter, or maybe also another hormone that will carry out a specific biochemical response. You find this with macrophages in processes of immune responses. We'll take a look at an example. As we continue, you'll see some specific example about this. Macrophages are responding to some invasion of some virus. And you have this autocrine signaling also at the early stages of cells of the embryo. You need to signal themselves to divide as quickly as possible, because at such stage of embryo development, you have a very fast development going on in the tissues. Similar thing happens with cancer cells, where you have cancer cell proliferation occurring by autocrine signaling, and it is what can result into the formation of tumors, because these cells will be dividing so quickly. And so you have such autocrine signaling, at a very fast pace in such disease condition in cancer. Then if you take tissues like cardiac tissues, the heart, and then the nervous system, the central nervous system, the brain, you also have cells that are connected by what we refer to as gap junctions. And across these gap junctions, you have ions getting across from one cell to the other. These ion channels are created by what we refer to as connexons. So you see, this you have a cell, a first cell on the left-hand side. You see the membrane there, and you have a second cell on the right-hand side. And in the membranes of each of these cells, you have the connexon. And the connexon is a hexameric protein. So if you pay attention, you realize that each of these connexons, it is being shown, is consisting of 6 proteins. So it's hexameric. And if you take just one of the proteins, it is referred to as a connexin. So in place of the O is I. So it's connexin for one protein. But if you put all of these 6 proteins together to form that channel in the membrane, we refer to that as a connexon. So these connexons in the membranes join. So 2 cells with connexons join to form the gap junction. And through these gap junctions, you can have exchange of ions and some other small biomolecules across these cells. So it's also a form of cellular communication in these cardiac cells, brain cells, and neurons, for very fast movement of substances and signaling. Then we also have what we refer to as the paracrine signaling. In this, you have a signaling cell. So you see a signaling cell at the center, which secretes some signaling molecules, and these signaling molecules are for signaling other cells nearby. So these cells are not far away, anywhere they are nearby, and the signaling molecules bind to receptors on these cells that are nearby. You find this with, for instance, the pancreas that secretes the hormone, say insulin, that will be acting on some cells that are nearby in its environment, to control metabolism of carbohydrates. So you have such paracrine signaling also as one of the ways that cells communicate. This is a major one that we will actually talk about, because after we establish this foundation, what we'll do is we'll take the endocrine system, because the endocrine system is where we'll find the major hormones that we talk about most of the time. And the endocrine system is that which has some specific tissues or glands. So what you're seeing here, so if you have a specific tissue or gland that is having some specific cells that we are referring to as endocrine cells, this endocrine cell secretes a signaling molecule, and that signaling molecule goes into the bloodstream and flows through the bloodstream to a different cell, so into a different tissue. So you have the signaling from one tissue flowing in the bloodstream to a different tissue in a different location altogether. And so it is in that tissue that you have the target cells that are going to respond to those signaling molecules, what we are referring to as the hormones. This is different from, say, paracrine, which is secreting and directly it binds to the target cells that are nearby. So you see that difference, that here you have the molecule that has a target cell that it will bind to. All right. Now we've seen those different types of signaling between cells. We've been talking about the receptor, which is on the cell surface, that will be interacting with the ligand. In our case, it is going to be the hormone. The hormone is a ligand. And I also mentioned that a receptor can be a protein, or it can be a glycoprotein, a glycolipid that is embedded in the cell membrane. Of course, you can have receptors that are inside the cytoplasm. Those are like proteins inside the cytoplasm. They are not embedded in a cell membrane. And so we have these types of receptors. The cell surface, which we also refer to as the transmembrane receptors. It is actually what is being shown here, because you see the cell and you have the receptor, which is embedded in the cell membrane, and a ligand is coming to interact with that receptor. And like I said, you also have receptors which are internal. So you have internal receptors inside a cell. Those are going to be in the cytoplasm, and those will be for ligands that can penetrate through the cell membrane into the cytoplasm to interact with these receptors inside a cell. And then another thing you need to know about receptors is that receptors are specific to specific ligands. So a hormone, say for instance, if you take, say, growth hormone will have its specific receptor. If you take a hormone, say dopamine, it will have its receptor. So you have different receptors that recognize specific ligands or specific hormones. So not any ligand at all will bind to any receptor at all, but they are specific to each other. Okay, that's also established. Now we can go into classification of the hormones, and that is going to help us to understand the behavior or the mechanism of action by which the hormones act. The first class of the hormones we are looking at is hydrophobic, which we also refer to as group 1 hormones. And that should quickly tell you, okay, we can do this group 1, group 2. The group 2 will be hydrophilic. If it's not hydrophobic, it will be hydrophilic. So we'll come to the hydrophilic, which will be group 2. Now, the hydrophobic hormones, as I said earlier, these will be able to penetrate through the cell membrane because you know the cell membrane is a phospholipid bilayer. It's a lipid. So if it's a lipid, then hydrophobic substances will easily dissolve through it. And you see that the group 1 hormones are grouped into steroid hormones, thyroid hormones, and calcitriol. And all of these are hydrophobic. Steroid hormones, of course, you know, the steroids, coming from sterols, having this, the steroid nucleus, is a lipid. So it can easily penetrate through the cell membrane. You have thyroid hormones. If you look clearly at these, thyroid hormones look very much as a derivative of the amino acid tyrosine. If you pay attention to it, because tyrosine will have that benzene ring with a hydroxyl group on it. But here you have 2 rings, but it's hydrophobic, the thyroid hormones. The calcitriol. Calcitriol too, if you pay close attention to it, you realize that it's having a similar structure like the steroid, except that the second ring is open. If you pay attention to the ring of the steroid, you see it's similar to that of calcitriol, except that the second ring is open. So that too is hydrophobic. So you have the hydrophobic hormones, which can easily penetrate through the cell membrane. Now, these hydrophobic hormones, examples of them are, say, the sex hormones. In females, estrogens and progesterone. These are secreted by the ovaries and the adrenal cortex. The progestins also by ovaries and placenta. Now, estrogens, of course, responsible for female sexual characteristics and a menstrual cycle. And you have the progesterone involved also in menstrual cycle and the maintenance of pregnancy. Androgens, so these will be in males. All of these, when you look at the structure, they are steroid hormones. So they will have that structure of the steroid. These androgens also secreted from the testes and the adrenal cortex at the anterior position of the kidneys, responsible for male sexual characteristics and regulating the process of spermatogenesis. And you have glucocorticoids and mineralocorticoids. These are secreted also from the adrenal cortex, from the adrenal gland, which is on the kidneys. And you have them regulating various metabolic and immune response processes. The mineralocorticoids maintain salt and water balance there. So you have some mineral metabolism, which is being regulated by the mineralocorticoids. Calcitriol also, you find it being, produced in the kidneys, and they are for the metabolism of, calcium ions So absorption of calcium ions from the intestine, kidneys, and the bone marrow. And you have uh, the thyroid hormones, triiodothyronine and tetraiodothyronine. These are secreted from the thyroid glands, from the neck region, and these promote general metabolic processes like metabolism of carbohydrates, proteins, lipids. They regulate all these processes. So these are some examples of those are the examples of the hydrophobic hormones. Now, we're looking at the mechanism of action of these hydrophobic hormones. As I mentioned earlier, hydrophobic, meaning that they can easily penetrate through the plasma membrane because the plasma membrane is a phosphobilipid layer. And you see the hormone, which is represented there as a triangle with H there. Perhaps we've seen this mechanism when we were studying, I think, the activity of maybe transcription, translation, maybe regulation of gene expression. So you see, we are looking at it again so that you can quickly relate it to that. So regulation of transcription, and you see that is the hormone that is able to penetrate through the cell membrane, and the receptor is inside the cytosol. The hormone binds to the receptor to have the hormone receptor complex, which then can pass through the nuclear pore and bind to a promoter region of a gene, activating that gene, and then that gene gets transcribed into a messenger RNA. The messenger RNA then comes out into the cytosol, where it undergoes a process of translation. A specific protein is synthesized, and that protein, of course, will be responsible for a specific biochemical response. Depending on which protein it is, if it is a protein kinase, for instance, it means it will be involved in phosphorylation of some other compound, which would trigger some biochemical pathway. Now, if we take the group 2 hormones, these are hydrophilic. Group 2 hormones are hydrophilic. And they are either peptide hormones or they are amino acid derivatives. So we have peptide hormones like insulin, glucagon, oxytocin, antidiuretic hormone. We'll take a look at these things much more as we go ahead. And you have also amino acid derivatives, which we also refer to as catechol-amines. Here, you must pay attention to realize that there's difference between peptide hormones and amino acid derivatives. And that is to say, if you say peptide hormones, what it means is that that hormone contains at least 2 amino acids. To have a peptide, you must have at least 2 amino acids that bind to give you a dipeptide. Then you have a peptide. You can have a tripeptide. You can have oligopeptide. Let's say up to about 20 amino acids in a molecule. We can classify that as an oligopeptide. Beyond that will be a polypeptide. All of these are proteins. But if you say an amino acid derivative, it's a single amino acid that has a modification to its structure. And when you look at the examples here, you see dopamine, norepinephrine, and epinephrine. Sometimes people say norepinephrine and epinephrine. And these hormones are actually the same as say epinephrine is the same as adrenaline, and norepinephrine is the same as noradrenaline. So these are synonyms that you should also be clear about. Now you realize that if you pay attention to these structures, you see that there are modifications of the amino acid tyrosine, or you may look at it as a modification of phenylalanine. In any case, phenylalanine gets hydrolyzed and you have tyrosine. Now, what you also see is that you have catechol, which is this six-sided ring with the hydroxyl groups there. And you see that it's just an addition of side chains with amine groups on it that gives you the structure of the hormones. And that is why the amino acid derivatives, are also referred to as catechol amines. Catechol amines. And you see dopamine, the difference between dopamine and norepinephrine, for instance, is just a hydroxyl group, which is there blue on norepinephrine. You don't find that in dopamine. In epinephrine, aside that hydroxyl group, you also have a methyl group, CH3, which is on the amine group. So this group of hormones are amino acid derivatives, and these are hydrophilic. They are water soluble. All right, now the hydrophilic hormones, we subdivide them. We can classify them into three subclasses. And the first class are those hormones that have what we refer to as the cyclic adenosine monophosphate as a second messenger. What does that mean? Now, bring your mind to this. You have the hormone being hydrophilic. If the hormone is hydrophilic, it means that it cannot pass through the cell membrane, which is a phospholipid bilayer. It cannot pass through it. You know the hydrophobic hormones, we saw the mechanism. The hormone passed through the cell membrane and went into the cytoplasm. But the hydrophilic hormones cannot do that because they are water soluble and the cell membrane is lipid. So the hydrophilic hormones bind to receptors on the cell surface. So these are the hormones that will bind to the transmembrane receptors. Now, when they bind to the transmembrane receptors, they send a signal. And that signal goes to a molecule inside of the cell to carry out the biochemical response. So that molecule inside of the cell that takes the signal from the receptor that the hormone is bound to is what we are referring to as a second messenger. So the first messenger is the hormone binding on the receptor on the cell surface, and the second messenger is inside the cell in the cytosol. It is that receives the signal and carries out specific reactions. And one of those compounds that act as a second messenger is cyclic adenosine monophosphate. So the group 2 hormones have the first group of hormones as cyclic adenosine monophosphate dependent. They use this cyclic adenosine monophosphate as a second messenger. And you have a list of them. You have the adrenocorticotropic hormone secreted by the anterior pituitary. In fact, if you look at all of these, you realize that adrenocorticotropin hormone, follicle stimulating hormone, luteinizing hormone, chorionic gonadotropin, thyroid stimulating hormone, endorphins and enkephalins, all of these are secreted by the anterior pituitary. They are all secreted by the anterior pituitary in the brain. And you have, what the functions there. The adrenocorticotropic hormone will stimulate the release of adrenocorticosteroids. In fact, when we take a look at the structure of the endocrine system, you come to realize that a hormone secreted in the brain will stimulate the secretion of another hormone, and you have that reaction continuing until you have the very final response in a particular tissue that should be having the response. You have the follicle stimulating hormone. In females, it stimulates ovulation and estrogen synthesis. In males, it promotes the spermatogenesis. Luteinizing hormone stimulates synthesis of estrogens and progesterone, and also responsible for ovulation in females. In males, the luteinizing hormone, again, is promoting androgen synthesis, male hormone synthesis by the testes. You have the chorionic gonadotropin, stimulating progesterone release from the placenta. So that is going to be in pregnancy. We will see that actually this serves as some biological marker for pregnancy tests. You have thyroid stimulating hormone promoting the release of the thyroid hormones, triiodothyronine and tetraiodothyronine. Endorphins and enkephalins acting as some natural pain relievers and some endogenous analgesics. In other words, they are responsible for response to pain. You have the antidiuretic hormone, which is secreted by the posterior pituitary, and this promotes some water balance. So absorption, reabsorption of water in the kidneys during filtration, urine formation. You have the antidiuretic hormone responsible for water retention there. And you have glucagon, which does the opposite of what insulin does. Insulin is a hormone which is to decrease blood glucose levels. That is to stimulate the process of glycogenesis. You have excess glucose molecules going into the synthesis of glycogen to be stored in the liver and in the skeletal muscles. But glucagon does the opposite. It rather increases blood glucose level by stimulating glycogenolysis and lipolysis. That is the breakdown of glycogen in the liver and in the skeletal muscles, as well as breaking down lipid storage as triacylglycerols in the adipocytes of the adipose tissue under the skin, all that to generate energy. Then you have the parathyroid hormone secreted by the parathyroid glands. This is to also regulate calcium metabolism, increases serum calcium by absorption of the calcium ions from the bone marrow. Calcitonin secreted by the thyroid gland, will do the opposite, will lower the serum calcium levels, meaning that it goes to stimulate the decrease, or it goes to inhibit the calcium ion uptake from the bone and the kidneys. Epinephrine, norepinephrine, we've seen that, adrenaline, noradrenaline, from the adrenal medulla, so from the adrenal glands, which are on the kidneys. And these, you have epinephrine will be, both of them, they are referred to as flight or fright hormones. That is when there is some danger. Adrenaline is secreted to sort of increase metabolic processes. Noradrenaline, same. So you either fight. If it's an invasion of the system, the body has to generate enough energy to fight that, or to run away from whichever danger that is coming at such a body. You have the epinephrine increasing heart rate and blood pressure. And of course, it promotes glycogenolysis, yes, breakdown of the energy storage in the liver and the muscle. And also, yes, lipolysis, breakdown of the lipids in the adipose tissue. So that is same as glucagon. It's doing similar things like glucagon. And norepinephrine, same thing, stimulating lipolysis in the adipose tissue. So you see, adrenaline or adrenaline are regulating similar activity as glucagon does. So that is for the hormones that have cyclic adenosine monophosphate as their second messenger. Now we can take a look at the mechanism of action of these group of hormones that have the cyclic adenosine monophosphate as a second messenger. Perhaps these two, we have seen it already, looking at perhaps when we're looking at again regulation of transcription or so. I don't remember. Maybe not your class. We see here again the plasma membrane. So keep in mind that it is still a hydrophilic hormone. And so that hydrophilic hormone cannot penetrate through the plasma membrane. So it will bind on a receptor, which is a transmembrane protein. It is embedded in the plasma membrane. So the hormone from the top interacts with the receptor. And that receptor is in complex with what we refer to as G proteins. We have these proteins that are bound to energy in the form of guanosine triphosphate. And you have some proteins also that are bound in complex with this GTP, alpha, beta, gamma subunits. And when you have the hormone binding onto the receptor, it induces the activity of the enzyme adenylate cyclase, which is bound onto that complex in the plasma membrane. And what will adenylate cyclase do? It will convert ATP to cyclic adenosine monophosphate. So the cyclic adenosine monophosphate is what we are referring to as a second messenger. It's just a conversion of ATP. By dephosphorylation, you have two phosphate groups removed from the ATP, which means you will be left with adenosine monophosphate. But the enzyme, adenylate cyclase, forms a cyclicized form of that adenosine monophosphate, what we are referring to as a cyclic adenosine monophosphate. And that second messenger only goes to bind to regulatory portions of catalytic protein, an enzyme that is represented, the catalytic parts of the enzyme, CC, and then the regulatory portion, RR. Think of it this way. You see, we looked at some processes of repression. So if you just look at the R as a repressor, which means when it is bound onto those catalytic portions, or they are inhibitors, they are repressing the activity of the enzyme. And when cyclic adenosine monophosphate binds onto that regulatory portion, it dissociates away from the catalytic portion of the enzyme. And that catalytic part of the enzyme now can catalyze the phosphorylation of a protein. That can be an enzyme. It's an enzyme, which is a protein. And just by phosphorylation of it, we have seen this to be a form of activation of biomolecules. So a protein, and you have ATP in that reaction, from which you have a transfer of a phosphate group onto the protein to have a phosphoprotein, which is an activated form of that protein. Let's take it to be an enzyme. That phosphorylated form, or the active form of the enzyme, will then give an ultimate biochemical response. It will go to catalyze a specific biochemical reaction, which will be the ultimate response that is intended. Right. Now, apart from that example we've looked at, we just talked about a hormone which is binding and having such an activity. We can look at it more specifically where we see epinephrine, norepinephrine. So these are hormones: epinephrine, norepinephrine, glucagon, tyrosine, glucocorticoids, thyroid-stimulating hormone, adrenocorticotropin hormone. You have growth hormone. These hormones act as activators. So these are the ligands. They act as activators of adenylate cyclase, meaning that when they bind onto a receptor, they will trigger the conversion of ATP to cyclic adenosine monophosphate. Now you see an inhibitor, the inhibitors of that process of activation that results into the conversion of ATP to cyclic adenosine monophosphate. You see insulin. Of course, insulin does the opposite of glucagon. So you see insulin there as an inhibitor. You also see niacin, one of these vitamin Bs that we saw, I think, when we were looking at the coenzymes. And then you see prostaglandins also acting as inhibitors in that process. So they are going to prevent the formation of cyclic adenosine monophosphate from ATP. And even you see that insulin also activates the decyclization of the cyclic adenosine monophosphate. So it converts that cyclic adenosine monophosphate into 5' adenosine monophosphate into its straight chain form. It serves as an activator of this hormone, phosphodiesterase, to break down that cyclic adenosine monophosphate into its uncyclicized form. Caffeine does opposite of that. Caffeine will rather prevent the cyclic adenosine monophosphate from becoming a straight chain structure. And why all of this? the formation of the cyclic adenosine monophosphate. It is linked now, cyclic adenosine monophosphate, now, you see, as a second messenger, now stimulates a protein kinase. It's a protein which is responsible for the conversion of triacylglycerol lipase in its inactive form into its active form. What we saw as phosphorylation, a protein phosphorylated to a phosphoprotein. That's what a protein kinase is doing to this triacylglycerol lipase B as an inactive form to triacylglycerol lipase A. That is the active form of the triacylglycerol. And the triacylglycerol is responsible for initiating the breakdown of triacylglycerols. So in the beginning, you have triacylglycerol, the first molecule, triacylglycerol. One free fatty acid is broken from it. You are left with a diacylglycerol. And then you have the diacylglycerol lipase will come in that reaction, remove the second free fatty acid. You are left with a monoacylglycerol. Then you have the monoacylglycerol lipase will then hydrolyze off the last free fatty acid, and you are left with just the glycerol molecule. So what you are seeing is the process of lipolysis. That's what we were looking at in the table before this, that you have some of these hormones, epinephrine, norepinephrine, glucagon, stimulating the process of lipolysis. So you see, the very final process as a biochemical response is lipolysis. That is the breakdown of triacylglycerols into glycerol and fatty acids. Right. Now, maybe another interesting thing you can say about this is the fact that you see caffeine, for instance, is preventing this process, cyclic adenosine monophosphate converting back to the linear form. Practically, you see that there are some caffeinated drinks, some teas that are promoted for weight loss. And it's because if you look at it biochemically, caffeine in those products will promote the formation of cyclic adenosine monophosphate, which means it will promote the process of lipolysis, the breakdown of fats, so that you say that the fats that are under the skin in the adipose tissue will be broken down, which will eventually result into weight loss. So that's a biochemical basis for which such products are sometimes manufactured and marketed. Right. Now we take the second group. That is the second subgroup of the hydrophilic hormones. The first subgroup we just finished looking at was using cyclic adenosine monophosphate as a second messenger. Now, this second subgroup is using phosphatidylinositol and calcium as the second messenger group. So the two of them act together. The phosphatidylinositol and the calcium, they act together in the biochemical response process. The examples you see here are thyrotropin, releasing hormone, secreted by the hypothalamus. And that is to promote the secretion of thyroid stimulating hormone. You have the gonadotropin releasing hormone, also secreted by the hypothalamus in the brain. And that also stimulates the secretion of follicle-stimulating hormone and luteinizing hormone. We have gastrin, which is a digestive enzyme secreted by the stomach, and it stimulates gastric hydrochloric acid and pepsinogen secretion. Cholecystokinin, also digestive enzyme, secreted by the intestines, the small intestines, and this stimulates contraction of the gallbladder, and that is to say, it is going to promote the secretion of bile from the gallbladder, which means it is promoting the metabolism of lipids, because the bile will come to act as an emulsifier of the lipids in the duodenum. And so when the lipids are emulsified, then you can have lipases now acting on these lipids. You have also the secretion of pancreatic enzymes that are also regulated by cholecystokinin. So you have these examples of hormones that use phosphatidylinositol calcium mechanism as their second messengers. All right, we'll take a look at how that happens. Now we see the structure on the left-hand side. That is phosphatidylinositol 4,5-biphosphate. Now, intentionally, what I've done is I have shaded specific portions. I've shaded a portion so that you can easily relate with the structure. Now, when we say phosphatidylinositol, that is without two phosphate groups. With this phosphate group on carbon number 5, and here the phosphate group on carbon number 4, which are in much deeper colors, much deeper shades. These two phosphate groups, if they are not there, the whole structure that you have will be phosphatidylinositol. But you see, if you have those two phosphate groups on carbon number 4 and carbon number 5, then you see the continuation of the name, 4,5-biphosphate. Great. Then you see that the structure, the whole structure is such that I have a glycerol backbone. So you see the glycerol backbone there. And on the glycerol backbone, you have fatty acids on these first two carbons. And that's this whole structure that is shaded in blue is a diacyl glycerol. Diacyl glycerol, meaning you have a glycerol backbone with two fatty acids on it. So you have a diacyl glycerol, that blue portion. If you take the whole portion down, the pinkish shaded square down, that whole structure will be the inositol 1,4,5-triphosphate. I mean, the structure you see, the 6-ring structure, is inositol, and you have phosphate group on carbon number 1, phosphate group on carbon number 4, phosphate group on carbon number 5. So altogether gives you 1,4,5-triphosphate inositol, or we say inositol 1,4,5-triphosphate. All right, so if that understood, we said the second messenger is phosphatidylinositol calcium. So working together, phosphatidylinositol and calcium acting together. Now what we see here is phosphatidylinositol. So that is if you have the hormone binding onto the cell membrane, the receptor in the cell membrane, that transmembrane protein, it triggers the phosphorylation of phosphatidylinositol. So you have two molecules of ATP coming in that reaction. So from phosphatidylinositol with two molecules of ATP, phosphorylation occurs to give you phosphatidylinositol 4,5-biphosphate, which is the whole structure that you are seeing there. Now, the next thing you see is that there's hydrolysis of diacylglycerol away from inositol 1,4,5-triphosphate. So intentionally, I've brought the structure here, and I've even explained the structure first of all, so that quickly, when you are revising, you can be referring to the structure and quickly understand that if you say hydrolyze diacylglycerol away to get 1,4,5-triphosphate, it only means you are hydrolyzing the blue shaded portion up away from the square portion down. So the blue portion up is what will give you that diacylglycerol, and the pink portion down there, the square, will give you the inositol 1,4,5-triphosphate. Good. Now, the inositol 1,4,5-triphosphate stimulates the release of intracellular calcium ions. So you saw we refer to the fact that you have calcium ions that can be released from the kidneys, can be released from the bone marrow. So inositol, the 1,4,5-triphosphate will stimulate that. And what do that calcium ions do? Those calcium ions act as cofactors to activate a protein kinase. So they will bind to a protein as cofactors, making those proteins now activated, the protein kinase. Either that, or it can bind directly to other enzymes, not only the protein kinase C, but it can also bind to other enzymes as a cofactor, and those enzymes will be active and produce an ultimate biochemical response. The diacylglycerol does the same, acting. We'll see a specific example where the diacylglycerol is, which is now activated by the calcium ions, so that it will now be fully activated to go into the regulation of other biochemical reactions by phosphorylating some proteins, because it's a protein kinase. So what it does is to catalyze phosphorylation. We've seen this type of reactions. I think we did something on proteins, and we talked about proteins as enzymes. Okay, so you have that. Let's take a look at it more colorfully, looking at that process of the phosphatidylinositol and calcium. So here you are. You have a cell membrane, which is green at the top there. So outside of the cell is on top of that green part, the green shaded portion, and inside of the cell, what is what we are referring to intracellular, inside of the cell, extracellular, outside of the cell. And what we see here is the structure, the phosphatidyl inositol. And this phosphatidyl inositol, upon the stimulation of a hormone, so a hormone binds onto a receptor that will activate, say, phospholipase C, and that phospholipase C will then cleave off the diacylglycerol away from the phosphatidyl, from the the square box. Let me go back to that structure. You mean this square box down here where you have the inositol triphosphate, the down part here. This is what we are looking at. So you have the phospholipase C, which is cleaving off that down part, which is now represented as the red circle, and that is the inositol triphosphate. And that inositol triphosphate acts as a ligand binding onto calcium channels. These are some transmembrane proteins that are embedded in the endoplasmic reticulum membrane. And upon binding onto that calcium ion channel, proteins, the ion channel opens. And when it opens, it releases calcium ions from the endoplasmic reticulum to the cytoplasm. And these calcium ions serve as cofactors. So the calcium ion binds onto the apoprotein of the protein kinase C that goes back to bind to the diacylglycerol. Remember the diacylglycerol from the beginning, from the phosphatidyl inositol. After the phospholipase C cleaves off the inositol triphosphate, you see the protein kinase C, which is now bound to the calcium ion, binding back onto that diacylglycerol, having now a full activated enzyme complex, and can carry out the process of phosphorylation of a substrate. That's a protein, phosphorylating it, or maybe some other biomolecule that will go into a specific biochemical reaction. All right. Now, we can take a look also at other hormones which fall in a third subclass. The first subclass was using cyclic adenosine monophosphate as a second messenger. The second subclass is just what we have finished looking at, which is using phosphatidyl inositol calcium as the second messenger. Now, the third subclass make use of different molecules as or different systems, some of them even a combination, as second messengers. We've looked at this already, which is the inositol triphosphates, diacylglycerol, calcium ion system. That's just what we finished looking at. This last one here is such a complex pathway. We'll not be looking at that. It's but whatever it is, they are just complexes of protein kinases. So it's the same things, but you have several cascade of reactions of phosphorylation of proteins that will result into phosphorylation of another all the way. But it's a whole complex web of reactions that can be clumsy. Well, if you are interested, you can just go online and look at it yourself, just to at least have a view of how it looks like. But we can take a look at this, the Jack-Stat system, as a second messenger. So we'll look at this Jack-Stat system as a third system in addition to those two systems that we have just looked at. And we have these examples of these growth hormone, prolactin. These are secreted by the anterior pituitary in the brain, the growth hormone promoting growth of the body. I mean, bone elongation and organs. And so you have the prolactin also regulating the growth of the mammary glands in females, and also lactation, that is after labor. You have prolactin, responsible for the production of breast milk. Oxytocin, secreted by the posterior pituitary of the brain, stimulating uterine contraction in childbirth, in labor, and also for milk ejection from the mammary glands. You have here insulin. So insulin is one of those in this subgroup. But I think we've seen insulin also acting. So you see, this is what I mean by you have some of the hormones having a combination of some of these systems, so that we've seen insulin using the system of the cyclic adenosine monophosphate, but in that process, insulin was acting as an inhibitor, or either it's acting as an inhibitor in the process of the conversion of ATP to cyclic adenosine monophosphate, or it was acting to convert the cyclic adenosine monophosphate into its linear form. We saw that example there. But here you will see insulin also making use of maybe for this inositol triphosphate system, for instance. So it's a combination of these things. And you have, of course, insulin is secreted by the pancreas. And it causes hypoglycemic effects, lowering blood glucose. That is to say, it's promoting the synthesis of lipogenesis. So if there are more lipids, it promotes those lipids going into the synthesis of the triacylglycerols that will be stored in the adipose tissue. Then we have this, what we refer to as somatomedins. These are some insulin-like growth factors secreted by the liver because they regulate growth by the growth hormone, which we saw results into, say, growth of bones and organs, of course, also for the growth of cartilages at the joints, for instance. So it's for general growth regulation. Now, the Jack-Stat mechanism as the third mechanism to add to the two that we have already learned. Now we see cytokine as the ligand, the hormone, which is coming to bind onto the cytokine receptor. And the cytokine receptor is a dimer. You see that that's a protein. It's a transmembrane protein embedded in the plasma membrane. And on it, you have some protein which is bound. So you have two proteins bound on. You have one protein bound on each of the monomers of the dimer of the receptor. And that protein is a JAK. That JAK is Janus kinase. So you have written it down here so that when you are revising, you can quickly refer to it. The JAK refers to Janus kinase. So it's some type of protein which is also involved in the catalyzation of transfer of phosphate group. So it's phosphorylation. Phosphorylation. Right. Now, the Janus kinase, after you have the cytokine now bound on the receptor, the Janus kinase catalyzes phosphorylation on the receptor. So you see, there's one phosphate group that has come to bind on the receptors, the protein there. And the second phosphorylation binds together with another protein, two other proteins that have come to bind there, and that is START. That START is also, it also means the signal transducer and activator of transcription protein. Signal transducer. So you can say a signal transporter. And at the same time, it activates transcription. It's a protein. The START is a protein. And what do you see? Now you have a full complex activated, and that activation results into dissociation of the START with two of the phosphate groups to form a dimer. That START becomes a second messenger that can move. So the first messenger is the cytokine that binds to the receptor on the cell surface. And you're having now a second messenger, a dimer, the START dimer, which now passes through the nuclear membrane into the nucleus and induces transcription of a specific protein. And of course, that transcription will continue. Transcription of a specific gene that will then result into messenger RNA that will get translated into specific protein to carry out a specific reaction. Let's look at it with a specific example. Say this is a macrophage cell. So it's a macrophage, an immune cell that is reacting to invasion of a coronavirus. And you see there is a receptor in the cell membrane of that macrophage that has attacked that coronavirus by the process of phagocytosis. So it engulfs the coronavirus. And you see the receptor there that recognizes that invasion of the virus, stimulates the process of the dissociation of the START from that JAK START system. And so you have the dimer of the START that binds to a promoter region of a gene. And the gene gets transcribed. And once the gene gets transcribed, it will get translated. And that translation, of course, is going to be some more cytokines that are produced. These are proteins. And these go out of the macrophage. And the cytokine binds onto the receptor of that macrophage. Of course, once you have more cytokines that are produced, they are also going to signal other macrophages and T helper cells. So different cells of the immune system. And you also see that the cytokine binds onto the receptor on the surface of the cell. This is an example of an autocrine signaling that we saw. So cytokine release from the cell, and that cytokine comes to bind on the receptor. And when it binds on the receptor, it causes activation of that receptor. So you see here, you see the JAK-STAT is bound onto the receptor upon activation. You have the STAT complex, the dimer, dissociating away, moving into the nucleus to bind to the promoter region. So that is a specific example that we can look at in terms of the JAK-STAT mechanism, in terms of second messengers for the hydrophilic hormones. Right. We'll quickly lay some foundation for our next lecture. When we come, we'll be looking at the endocrine system. And that is going to be looking at all these glands that are responsible for the secretion of specific hormones, glands or tissues. So we have the pineal gland, the pituitary gland. These are found in the brain. Of course, even above this, we'll talk about the hypothalamus. These secrete some specific hormones. And then we see the thyroid and the parathyroid glands in the neck region, also secreting some specific hormones. The adrenal gland. So you have these adrenal glands, which are at the anterior position of the kidneys, which also secrete some hormones. We have the pancreas, also secreting some hormones like insulin. You have the ovaries in females, the testes in males, responsible for the secretion of sex hormones. As the general system, the endocrine system that we'll look at when we come the next time. And then we will take a look at each of these hormones that are secreted by the various glands and tissues that form the endocrine system. So we'll see hypothalamus, which itself not necessarily a gland, but it's referred to as a master gland because it is that which actually releases what we refer to as releasing hormones, having a direct stimulation on the anterior pituitary. So hypothalamus secretes thyrotropin releasing hormone, corticotropin releasing hormone, gonadotropin releasing hormone, growth hormone, and growth releasing hormone, and then growth releasing inhibiting hormone. Then we have the prolactin releasing inhibiting hormone. All of these acting directly on the anterior pituitary. And their activities follow. So thyrotropin releasing hormone will stimulate the anterior pituitary to release thyroid stimulating hormone. And that thyroid stimulating hormone will then stimulate the thyroids to secrete thyroid hormones, triiodothyronine and tetraiodothyronine. Similarly, you have the corticotropin releasing hormone acting on the anterior pituitary to secrete the adenocorticotropin hormone, which will then act on the adrenal cortex to secrete adrenocorticosteroids. You have the, again, hypothalamus, which releases gonadotropin releasing hormone, which will act on the anterior pituitary to secrete luteinizing hormone and follicle stimulating hormone. These two act on the ovaries and the testes. The ovaries then secrete estrogens and progestins in females. In males, the testes then produce the male sex hormones, androgens. And these will act on the reproductive system. And you have the growth hormone releasing hormone, the growth hormone releasing inhibiting hormone. These two act together to regulate growth, so that you have this acting on the anterior pituitary, regulating the secretion of growth hormone from the anterior pituitary. And that growth hormone can act directly on bones. So in children, it will be resulting into growth, say elongation of bones. Of course, other tissues, the growth of those tissues. The growth hormone also acts on the liver to release somatomedins, which we saw much earlier, also regulating growth. Then you have the prolactin releasing inhibiting hormone. So this interesting mechanism here is that the anterior pituitary releases prolactin not under the regulation of the hypothalamus. What I mean is, it's not the hypothalamus that stimulates the release of the prolactin from anterior pituitary. It's rather the opposite, that the hypothalamus rather secretes prolactin releasing inhibiting hormone to inhibit that secretion. It's a form of regulation so that you don't have too much of the prolactin being secreted. And prolactin acts directly on the mammary glands in females. I mean, in milk production after childbirth, you see that. You see the posterior pituitary is not under the regulation of the hypothalamus. It's autonomous, it's on its own. And this secretes the antidiuretic hormone and oxytocin. The antidiuretic hormone regulates water reabsorption, say water balance in the body, the kidneys. You have the oxytocin also responsible for uterine contraction there. Then generally you see that the hormones, after they are secreted in adequate amounts, act as inhibitors to the anterior pituitary. That is giving a feedback. So by feedback mechanism, the secretion of the hormones from the anterior pituitary and the hypothalamus are regulated. The anterior pituitary also gives a feedback mechanism to hypothalamus, so that if you have hormones that are secreted by the hypothalamus in adequate amounts, it gives that feedback to inhibit further secretion of those hormones. So this is the general structure of the endocrine system. When we come next time, we'll take a look at each of these hormones, I mean the glands, each of the hormones and what they are doing. We'll take a look at the structure and their function, activity, mechanism. We'll take a look at those things as far as the biochemistry of them is concerned. Raymond Opoku And Nancy Ajay, they say, please, which hormone also stimulates the testes to release testosterone? So the androgens, when I say androgens, it refers to, the male hormones in general. But this is a foundation. I think that we shouldn't jump the gun. When we come next week, we will go into the details of each and every one of these ones. So let's not jump the gun. Today we just laid a foundation. We looked at classification, first mechanism of cellular communication, and then we looked at the various classifications and the mechanism of action of the hormones, looking at the second messengers for the hydrophilic hormones and all of that. But if you start talking about specific hormones now, it's like we are jumping the gun. So you hold it. When we come, we'll take a look at them all. All right. Raymond says then it's very deep. Yes, it's good to dig deeper than just to be digging shallow. All right. So I think we've done justice to the plan we have for today. Now the floor is open. If you have any questions, if you have any contributions, anything you would like to say, I'm all ears. I will, you just raise your hand and I will enable your microphone. You unmute yourself and speak, or you write in the chat and I will respond. The floor is open.